This video provides a short introduction to this Visium data analysis course. So first we look what is spatially resolved transcriptomics, how the Visium system works, what you should keep in mind when working with this kind of data, and then finally what will you learn during this course. So spatially resolved transcriptomics provides the spatial context for gene expression data. In other words, we can overlay the gene expression data. In this image here, we have clusters um, with a tissue image. So this retains the organization of tissue and cellular microenvironment as opposed to single-cell RNA-seq data where you actually need to dissociate the cells from each other. So location matters for many applications. For example, being able to identify cell types in the context of heterogeneous tissue is important for cancer studies. There are several technologies available to produce this kind of data, and our course focuses on analyzing the next genomics Visium data. So how does the Visium platform work? First, a tissue slice, either frozen or FFPE slice, is placed on a capture area on a slide. This capture area contains about 5,000 spots, and the spots are barcoded. So the diameter of a spot is 55 micrometers, and the distance between the center of a spot to another center is 100 micrometers, meaning that there is some empty space in between. Important thing to remember here is that depending on the size of the cells in your sample, there are typically between 1 to 10 cells per spot, so not just one cell, but several. Let's have a look in more detail at these spots. So they contain millions of capture probes. Each capture probe has a spatial barcode, which helps to track back from which spot the transcript came from. Then there is the unique molecular identifier, or UMI, for counting the transcripts, and then a poly-DT for capturing poly-A RNA. So when the slice is placed on this capture area, it is first stained and imaged, then the cells are permeabilized, then a reverse transcription is performed, cDNA synthesis is then followed by library construction, and finally sequencing. So what you typically get from your core facility is then a table where you have gene expression values for each of these spots, and additionally information where from which location a particular spot came from. So things to remember is that each spot typically includes several cells, not just one. Those cells in a spot can be of different types, so not just one type, but a mixture. So consequently, the gene expression values that we get are actually an average from the cells in a spot. And therefore, the clusters calculated based on these values represent a group of spots with similar composition of cell types. During this course, you will learn how to create a cell route object from Visium data and how to check the quality of spots. You also learn how to filter out low-quality spots containing damaged tissue. You will then normalize gene expression values and identify highly variable genes, which are used for principal component analysis in order to reduce dimensions of the data. The principal components then are used to cluster the spots with graph-based clustering. And then we visualize the clusters both using the UMAP method, and we also overlay the clusters on the tissue image. Then we detect spatially variable genes and visualize them on the tissue image. You will learn how to subset out anatomical regions or clusters. And importantly, you will learn how to predict cell type composition in a spot. So for this, we use integration with single-cell RNA-seq data. So single-cell RNA-seq data here is used as a reference. And finally, you will learn how to combine several samples.